Okay, so welcome everyone. Thank you for coming. We have 11 people online today as well. So, oh, no, 12 now. If you're here in person, please sign in on the sign-up sheet at the front of the room. If you're online, please type your name into the text chat to everyone so that the text chat is automatically recorded for the session. So that way I'll know who's online. Excuse me. So today I'm going to be talking about high throughput technologies. And the reason why I give this lecture, there's not a lot of bioinformatics pipelines or analysis in it, but understanding how the different technologies work and where your data comes from can influence your pipeline. So that's the main goal of today. When someone comes to me with a with a new data set, the first question I always ask, well, there's multiple questions that I always ask about how the data was generated, what technology was it on, and specifics that the sequencing facility can actually give me. And that influences how I go about building my bioinformatics pipeline. So the learning objectives for today, I want you guys to be able to describe the basics of how microarrays and sequencing technologies work. Also to be able to discuss the advantages and disadvantages of microarray technology versus sequencing, and understand how sequencing technologies can actually be complementary to each other. There's not one technology out there that's perfect for all problems. And most importantly, I want everyone to be able to list a few examples of how the technology and use of the technology can influence the bioinformatics pipeline downstream. And so the quiz questions are actually based off of these learning objectives. First, a little bit of introduction. This may be review for some of you. The central dogma of molecular biology gives that your DNA, which contains all the instructions and building blocks for life, gets translated into transcribed into RNA, which is then transcribed into proteins. And proteins are actually the workhorses of your body, and they're the ones that do a lot of the work. So my lecture is focused most on gene expression, and that is the abundance of messenger RNA in the system. Your gene expression is very dynamic and different for each system. For example, in the immune system, your dendritic cells float around looking for pathogens. When they find one, that signals a whole signaling cascade and results in massive gene expression changes throughout the cell to cause the dendritic cell to go through physiological changes into mature dendritic cells, which then look different and also behave differently. Also in cancer, the gene P53 is a tumor suppressor gene. When this gene is perturbed, it causes a lot of downstream gene expression changes in multiple systems, including the cell cycle and cell death, which can result in cancer. Another example is the circadian rhythm. There's daily gene expression changes. Certain genes are turned on and turned off throughout the day with the circadian rhythm. So those are just a couple of examples of how dynamic gene expression can be. So to measure gene expression, you can use microarray technology or sequencing. In this lecture, I'll cover Affymetrix in a little bit of detail and then talk about Agilent. And then sequencing technology, I'm going to focus on 3 next generation or second generation sequencing technologies. You also have first generation and third generation technologies, which are coming more to the forefront, but I haven't had any personal experience with those. So what are microarrays? Microarrays actually give you the capability of measuring thousands and thousands of genes and their level of expression in parallel. So it gives you a global snapshot of the gene expression or transcriptional state of whatever system you're studying at that time. So with that, you can actually compare the global gene expression of different systems. For example, before and after intervention or treatment, across cellular tissue types, such as compare the gene expression between normal tissue and cancerous tissue, or over time. For example, the dendritic cell maturation takes about 24 to 48 hours, and you can measure how the genes are changing over time with microarrays. You can also map genes to phenotypes and identify genes that control other genes, such as doing a knockout experiment. If you knock out the expression of a particular transcription factor, then you can measure the gene expression before you knock it out and then after it's knocked out, and look at which genes change expression to get an idea of which genes are in that transcription factor's control. So microarray terminology, to start, a probe is a defined sequence of nucleotides that are attached to the array surface. So microarrays are glass slides, and you have little spots of these known sequences. So you have to know the sequence of the genes that you're targeting to build these probes. And they're about 25 to 60 base pairs long, depending on which technology you're using. The target is your unknown sequence of nucleotides in your hybridization mixture. So your sample that you're trying to measure and figure out which genes are expressed in your sample, those are considered the target. So when your sample is mixed over the Affymetric or the microarray slide, it's called hybridization, and it's a sequence-specific interactions between two complementary strands of nucleic acids. So with DNA, guanine always binds with cytosine, and adenine always binds with thymine. And so it's very sequence specific. So in that way, these areas of the probe can target specific genes that you're looking for. Okay, so here's just a little workflow for microarrays with a cancer example. So you have your normal tissue and your tumor tissue. You extract out your RNA, and then you actually label these with fluorescent dyes. So your normal in this example is green. Your cancer sample is labeled red, and then you hybridize it to your array. And then you scan that array with green or red lasers to get out the fluorescent intensity of where your cancer samples have hybridized to, or your normal samples. And then these two are computationally combined to get one image that looks something like this for both samples. So here, if you have genes that are expressed only in your normal sample, those cells are going to appear mostly green. If you have genes that are expressed only in your cancer sample, those cells will be mostly red. So, and then if they're expressed in both, it's gonna look mixed or yellow. So here, anything that is, since our normal is our control, any gene that's only expressed in normal and not expressed in cancer, we can say has been downregulated or turned off in the cancer cells. On the other hand, if it's only expressed in tumor and wasn't originally expressed in the normal, we can say it's upregulated in the cancer. So this is actually called a two-color array because you have two samples on the same array and each are labeled with a different color. And the key thing to remember about two-color arrays are that the values that you get out are always relative to the control that you put on that array, and you can't change the control that you're using. So, for example, you would only have the normal sample that these values are relative to with that tumor sample. On the other hand, you can also do one-color arrays, where you only have one sample per microarray. And the advantages of this are the gene expression that you measure is the signal intensity that you get from those labeled dyes, and it's not relative to a control. So you would have your normal on one chip and your tumor or your cancer sample on another chip, and you'd be able to compare those after the fact using your bioinformatics pipeline but that wouldn't be the only comparison you could make. If you had multiple samples, then you could make multiple types of comparison. For example, we have, in one project I'm working with, we have normal mouse tissue, and then we have PDX, patient-derived xenograft data from the liver and the brain and the lung. So we can make multiple comparisons and compare the brain to the liver, or the liver to the lung, or any of those to the mouse, to the normal. So one color arrays give you much more flexibility in the comparisons that you can make. The one disadvantage is that you have to use more arrays, but on the contrary to that, you might have to use more arrays, but then you could also par down your normals. So instead of having a normal sample for every cancer sample that you're running, you can have 3 normals and then however many cancer samples and compare them all to your smaller normal set. So that could save there. But the big message here is that your downstream analysis can be different for data from 1 to 2 color arrays. So you need to know where those numbers in your Excel sheet are coming from in order to do the analysis well. Affymetrix is one company that does 1 color arrays. And you don't need to know all the details about how these arrays are made, but I looked them up, and they use lithographic masks. And the main thing is it's not cheap to make these arrays. There's a long, complicated process that they have to go through to make one Affymetrix gene chip. And so they make templates to make the Affymetrix chips. And so this makes it difficult to customize, and which results in the majority of Affymetrix arrays are highly standardized. So once Affymetrix creates an array for, say, human or mouse, everybody uses that same array. It's got the same genes on it in the same locations. And so this makes it easy to go onto public databases like Geo and look for Affymetrix human array and be able to compare your experiment to their experiment because it's the same chip. Okay, this is one of a few down the rabbit hole slides. So if you see this little symbol up here, that just means that it might be a little more detailed than what you need. So, but I find this is some helpful information for people that work with Affymetrix. So one peculiarity about Affymetrix is that it measures 1 gene with multiple probes, where other technologies measure 1 gene with 1 probe. So with Affymetrix, 1 expression value in your matrix for a gene expression actually comes from about 11 to 20 different summarized values from a set of probes called a probe set. And these probe sets are actually scattered all around the chip. And what that does is helps normalize out some of the noise. If the hybridization mixture didn't mix across the whole chip evenly, you're actually getting samples from the whole chip instead of just 1 location. And then if that 1 location gets messed up, your whole measurement for that gene gets messed up. So just to keep in mind that when you're analyzing Affymetrix data, if your data looks weird or off or unexpected, you can actually drill that back down to this probe set level and do your own summarization. Generally, this is all done with the Affymetrix software, so you'll get an Excel sheet with 1 value. But just know that that's actually summarized across 11 to 20 different measurements for that same probe. Another peculiarity about Affymetrix is its many-to-many relationships with probe set IDs. So if you've ever tried to take probe set IDs and map them to gene names, you might get a bit of a surprise for some of them, because multiple probe set IDs can actually map to 1 gene, and then also 1 probe set could actually cross-hybridize to multiple genes in the same family. And it does this because of how they design each probe. Again, each probe is about 25 nucleotides long. And so that limits it to where there might not be that many unique places of 20, 25 base pairs in the genome to target one specific gene only. So on the previous slide, down here, all of these little sequences here are the different probes that probe this one gene, and so a lot of the Affymetrix arrays only probe the 3' end of the gene. So you're not even sampling the entire gene, only a small part of it. And so some of these short sequences can actually are identical to other genes in the same family, so it's really hard for to target some genes precisely. You can tell a lot of this by the _X or _S on the Affymetrix probe ID, that it might not be unique. So that's important because when you're interpreting your results and all of your probe sets for a gene are known to cross-hybridize with other genes of the same family, you might need to go do further experimentation to make sure it's the gene that you're actually interested in. And not one of these others, or it's a mixture of genes that are hybridizing to that one probe set. So there are also various ways to summarize a probe set, and there are a few different normalization methods for Affy arrays. And when you look through the literature, there's papers that directly contradict other papers, and I just wanted to say that they may not be one particular normalization method or probe set summarization method might not be generalizable to all applications. For example, this paper here recommends that the GC-RMA normalization is best for identifying differentially expressed genes, and they did an experiment to show that. Whereas this paper here indicates that the raw data from microarrays might be better for identifying phenotypes in cancer patients rather than the process normalized data, and they have experiments to show why. So the point is to just, when you're building up your bioinformatics pipeline, be aware that there are different methods that may be more applicable to one type of experiment or biological question that you're trying to ask than another. All right, Agilent microarrays are different from Affymetrix in that they actually use HP inkjet technology, and they print the probes on the array. This makes the arrays very cheap to construct, which makes them very easily customizable. And Agilent also offers 1 and 2 color arrays if you're interested in the 2 color arrays. So Affymetrix versus Agilent, again, it depends on what questions that you're asking. Affymetrix is more standardized. There are a lot of Affymetrix experiments out there. But if you're looking for a specific set of genes for your question that you're asking, then you might want to go with Agilent if there's no array out there that targets that set of genes. So Affymetrix is not customizable, Agilent is. Affymetrix has the probe set, so again, one value is summarized by multiple measurements from the same probe. And Agilent just has 1 probe per gene. Also, Agilent actually has a longer probe, so they're 60 base pairs long, or 60 bases long, not base pairs. So you're able to find more unique targeting of your genes of interest with that longer probe. So the overall message is just know the composition of what technology your data was based off of so that you can use that to either help analyze your data or help figure out and problem solve any oddities that come up in your analysis. So now we're going to transition into sequencing. Comparing microarrays to sequencing, the upside is microarrays are very affordable. They've been around for a long time, so the analysis is straightforward, and there are a lot of tools out there to help you analyze and process microarray data. and then also has very low storage and computational requirements. So microarray experiments can be processed on a laptop. The downside is you are stuck with what is on the chip, so you have to know what you're looking for before you can measure it. And probes can cross-hybridize, so that can cause noise in some experiments. And you can't view the actual fragments from your sample that are binding to the chip. You can only see the signal intensities, which could be a mixture of multiple genes if it's a probe that cross-hybridizes. So sequencing actually overcomes many of these limitations of microarrays, but it introduces a new set of challenges. This is just a summary of my take on RNA-seq versus microarrays for gene expression analysis. RNA-seq actually measures all of the genes on the chip, not just the ones that you know, so you can discover novel genes. It actually samples the full transcript, not just your small 25 or 60 bases on your probe. And for each read, which we'll learn about in a minute, it's actually longer, up to 125, or technologies are even getting up to 400 or 500 base pairs that they can measure. And they're somewhat more sensitive to low expression. And the two red yeses in the RNA-seq column, I just put those in there this year. They weren't there last year because the methods to process sequencing data are becoming more and more standardized over the years. You start to see the same pipelines and publications. And so the bioinformatics part is becoming, I think, more standardized. But you still have tons of data. You're not going to get around that. So you still need a server or a cluster to process sequencing experiments. And the bioinformatics analysis and quality control and debugging can be more complex. Okay. Do we have any questions so far on microarrays? Okay. A few quick slides on what sequencing is in general. It's determining the nucleotide order of the DNA or your RNA molecule, whatever you happen to be measuring. And so you have your large molecule, and then you break that up into small fragments, and there's a lot of different ways to do that. And then those are sequenced, and you get your computational sequences out, which then have to be aligned to your genome. And that's computationally intensive. And so in my RNA-seq lecture, I go into these steps here in a lot more detail. In this lecture, we're going to talk more about template preparation and sequencing and how choices made in these steps can influence how you analyze the data in the bioinformatics pipeline. So the general sequencing pipeline is you do template preparation, where you take your sample and you isolate the parts of it that you want to sequence and make lots of copies and put it on a slide. And then you do your sequencing, which determines the order of the bases. And then once you get your digital sequences out, then you can go into the bioinformatics analysis. So some terminology to go along with this is your biological sample is called your library. And I have this little note here. Your insert size is actually determined in your library preparation steps. And I'm going to talk about insert size in a few minutes with respect to bioinformatics pipeline. Once it's sequenced, you get out your digital sequences, which we call reads. And then those digital reads are then aligned to a known reference genome. So we have various versions of the human genome, of mouse genome. There's hundreds of them, thousands of reference genomes out there. If you don't have a reference genome, so if you're sequencing viruses or novel virus, and you don't have a reference genome for that, you can do de novo assembly, which just takes all your reads and tries to overlap them, like trying to put a puzzle back together. And it will create a reference sequence for you. So one important terminology to go along with sequencing is coverage. There's two types of coverage that you'll see in the literature. The first is depth of coverage. So here we have our genome, and this is just an annotation for a gene, where the black bars are your exons, or the parts that actually get translated into the protein, and the thinner bars are those that are spliced out. Here, each of the little blue lines is a read from your sequencer, and depth of coverage refers to, given any genomic position, how many reads actually align to that position. So for example, if 60 reads align to a given position, that's termed 60x coverage. That's your depth of coverage. Breadth of coverage is the other way. How many base pairs are covered by at least X amount of coverage. So if you're looking at 1x, so you want at least one read to align to a base pair, then you can say 80% of my genome was covered by at least one read. So sometimes you'll see in the literature, we have 80% coverage at 60x. That means 80% of the regions that they were interested in were covered by 60 reads depth. So this 60x can be a little tricky sometimes. They'll mention in the literature or not. It could be your 60x minimum, maximum, or average. A lot of times it's average. So if they say 80% coverage at an average of 60x, you can still have some part of that 80% being covered by less than 60x. And that matters because when you get into bioinformatics pipelines that do variant calling and stuff like that, you actually need a lot of depth of coverage in your area of interest to be able to statistically detect those variants and mutations. So it's important to know what your coverage is. And it's also important to know what your target coverage is. Because if the sequencing facility doesn't give you back your target coverage, you can generally have them rerun your sample again at no charge, generally. Because if you don't get your target coverage, you're not going to have enough power to be able to do some of the analysis you might want to do with your data. Multiplex sequencing is just a term I wanted to introduce. Multiplex sequencing allows the facilities to run multiple samples on the same lane of the sequencer. And this helps to reduce lane or chip-specific effects. So just like microarrays, maybe the mixture didn't wash across the whole sequencing lane evenly, or you get a bubble in it or something. So you can actually take your sample and mix it with other samples and distribute it across multiple lanes, which helps reduce those noisy effects. So with multiplex sequencing, they actually take your sample, which is here in the blue, and they add specific barcode DNA sequence to either end. Then they go ahead and they sequence. They mix all of those together, so your samples are mixed with a barcode. Say tumor has one barcode, normal has another barcode. They'll mix tumor and normal together, sequence them all on the same lane, on the same chip. And then once we get the digital reads out, they're able to look at those barcodes and digitally separate your samples in the post-processing. And then the sequencing facility usually does this for you, and they'll take off your barcode, the barcodes. However, sometimes you'll still get barcode contamination in the files that you get back. And so that's part of the quality control that you have to do in your bioinformatics pipeline, is look and check to make sure they remove the barcodes. Because one time I had a sample that the barcodes weren't removed. So you have to look and see if those barcodes were removed. And if not, you can remove them or have the sequencing facility remove them. Paired-end sequencing is another term I wanted to introduce. You can have paired-end sequencing or single-end sequencing, and it's important to know the differences between what each of these are in designing your experiment. So single-end sequencing, so this blue part here again is the fragment that you're interested in sequencing. Single-end sequencing will sequence from one end only up until however many base pairs the technology can do. So for Illumina, generally I get 125, 150 base pairs. But your actual fragment that you selected for may be 1,000 base pairs long. So you're only sequencing up to 150, say, base pairs of your actual fragment. Paired end sequencing actually does it in 2 rounds. First, it'll sequence from the one end, so it gets one end of the fragment, and then it does another cycle, and it will sequence it from the other end. So if you have, say, a fragment that's 500 base pairs long and your reads are 150, then you'll have 150 from this end and 150 from this end. And then you have about 200 base pairs in between that you're not actually sampling. What this does, having paired ends, is it helps the aligners in the bioinformatics pipeline. It'll help the aligners find a more exact position for where that fragment came from. And so I mentioned insert size. So the length of your fragment that you're targeting is actually your insert size. And that's determined in your library preparation steps. So usually I have to go back to the lab or the sequencing facility that processes the samples and say, what is the insert size that you targeted? Because once you chop up that DNA, they can actually go in and only pull out, say, sequences that are 1,000 base pairs long, 500 base pairs long. So you can do size filtering in the library prep step. So if I know what this insert size is, I can feed that into the alignment algorithms, and they can find better, more accurate alignments with the paired ends. If you don't have paired ends, then you don't need to know your insert size. Oh, and the paired ends also help with de novo assembly when you're trying to piece together your genome from scratch. It gives you a lot more information. For example, paired end sequencing on normal mapping, if this is your reference and this is your sample, and there's 1,000 base pairs in between each end, then you're going to expect to see 1,000 base pairs in the actual alignment in the mapping. But if you see more, so maybe there's 2,000 base pairs when you map it to your gene, when you map it to the reference, then that might actually mean that your sample had a deletion in between those. And the opposite for the insertion. So paired ends can actually give you whether or not there's deletions or insertions in your sample. So again, just more, paired ends give you more information. It is more expensive to run paired end sequencing. Okay. Are there any questions so far? So now we're just going to get into 3 of the technologies, Illumina, Solid, and Ion Torrent. The first step is always your template preparation. So each of these technologies does it a little differently. I'm not going to go into all the technical details, but what I want to mention is that in order for you to sequence your sample, you do have to have a lot of copies of your one single fragment. And so this is where they do PCR amplification, where it takes the fragment and it copies it over and over and over again. For example, in Illumina, that uses bridge amplification, you have your single fragment, and it'll bind to the chip, and then they'll copy it, and then you have 2, and they continue, and then until you get spots of hundreds of your single fragment that you're trying to sequence. And then that goes into the sequencer. But what can happen is this amplification can go crazy and create multiple spots of that one fragment that you're trying to sequence, which turns into multiple identical reads in the end. So when you're doing bioinformatics analysis, depending on what your question is and what type of sequencing you did, may require you to remove these PCR duplicates. So you have to actually analyze your file and figure out which reads are most likely duplicates and remove them. For example, if you're doing variant analysis, you have to remove PCR duplicates because it messes up the statistics of identifying variants. So next, after your template prep, then you go in and you do your sequencing. The first one I'll go over is Illumina HiSeq. Illumina does sequencing by synthesis. So it requires DNA polymerase and dye-label nucleotides. So each nucleotide is labeled with a different color. So each pass, they have your sequence that is attached to the slide, and each pass, it attaches one nucleotide, and then it has this stop on here so that it can't actually, the polymerase gets knocked off, so it can't actually add any more except one. It can only add one nucleotide at a time. So you're essentially synthesizing an exact copy of your fragment this way. So every cycle adds one more, and each cycle you take a picture and you figure out which color is in which spot. So here you can see for this particular fragment, the color changes for each cycle. And that's how you build up your digital sequence. The one thing to realize with this type of sequencing is the concept of dephasing. So the for each run, a nucleotide may or may not get added to all of your hundreds of fragments that are in one particular spot. Not all of them will have a nucleotide added. So the longer your read gets, the more likely it'll go from everyone's in sync, so it's showing the same color when you scan the chip, to all your reads are now out of sync with each other. So the sequencer does not have as much confidence as to what base was actually next. So this is why some of the technologies can't just sequence forever. They have their limits depending on the technology. This is just important to understand because in your bioinformatics pipeline, you're going to want, there's quality scores that go along with each base that's read. And you're going to want to look at those quality scores and trim your reads. If the quality is really bad, you're going to want to get rid of those base pairs from your read so that your aligners can do a better job of aligning that fragment to the genome. All right, solid. I've personally never worked with solid data, but from what I understand, debugging it can be different. So solid actually sequences by ligation. So instead of adding one base pair at a time, we now have these pre-made dye-based probes where you know the first two bases, and then the remaining 6 are bases that combine with anything. So they're anonymous bases. And so what they do is they go in rounds and they ligate these dye-based probes to your fragment, and you'll get something like this out where you'll have, it also measures the color sequence, but you'll notice not all the bases are interrogated during any one cycle. So what they do is they actually shorten their primer by 1, and so it ends up moving down 1 base pair each time. So this process is slower than Illumina, and they can't get the length that Illumina can. But one of the advantages of this is that each base is actually sequenced twice. So solid sequencing has higher accuracy than Illumina or other technologies. Some of the bioinformatics challenges are you have this color space where you have multiple rounds with different colors that have been measured for each fragment, and you have to actually combine those in the right order and then decode them in order to get your sequence out. So sometimes you'll see solid, color space. It's not actually the base pairs. You're actually seeing the sequence of colors, not base pairs. So you have to be able to decode those. And in order to do that, you have to know what your very first base pair was. If you don't know that for each fragment, then there's no way you can decode the rest of it. Generally, I believe the sequencing facilities will do all of that for you. There is software to do that. But just knowing about it when you're dealing with solid data is good in case you run into any problems. And this is just another slide with more information on how the color space is decoded into your base pair space, through your base space. So solid is definitely down the rabbit hole of sequencing. The last technology I wanted to cover was the ion torrent. And ion torrent requires DNA polymerase, so it is by synthesis. However, there's no labeled nucleotides. You don't have to label your nucleotides and process those. What it does is it detects the change in pH when a base is added. So you'll run, for example, all thymines over your chip. And if it detects a pH change for that spot or that probe, then it'll register that, oh, a thymine was added here, the pH changed. And what's really cool about this is you can actually detect multiple nucleotides added at the same time. So if you have a sequence that has 3 thymines in a row, this will actually add those 3 thymines in a row, and you'll measure out 3 times the pH change than if just one base pair was added. So this can actually, the ion torrent actually can go for longer before it dephases, so you get much longer reads out, but it is very slow and it's got low throughput. So this is just the slide that I've been showing up. Read lengths, again for ion torrent, are, I believe it's over 400 base pairs now. This is a little outdated. ABI solid is about 85 to 100 now, I think. And Illumina sequencing, I see 50 base pairs sometimes, but generally it's 125, 150 that I see on a regular basis. But Illumina has technologies that can create longer reads. Illumina by far is the cheapest, but it takes about 3 to 10 days, which is okay. And whereas ABI solid takes up to 2 weeks. Ion torrent is the fastest. Ion torrent is used for a lot of in-lab research, such as quickly sequencing new viruses that come out, where you need to know the sequence of that virus. And the virus's genome is much smaller than humans, so you can actually do that on an ion torrent very quickly to be able to characterize new viral strains. So it's used a lot for that. Throughput, if you need a lot of information, Illumina gives you the highest throughput at 600 gigabases or more. ABI solid is lower, and ion torrent is the lowest, but again, because it's small and can be run really fast and gives you long reads, it is good for certain use cases. And here you'll see the accuracy. Solid, again, is got the highest accuracy, whereas the other two are around 98%. So I just wanted to emphasize with this that these technologies are complementary to each other. There's no one technology that is perfect for all applications. Illumina HiSeq seems to be the most widely used. That's what I'm most familiar with. It can be used for almost any application. It's very versatile. However, if you're really interested in a specific region, for example, if you have a variant that you're interested in a particular region and you want to get a really accurate reading of is that variant there or not, such as a rare variant, you might want to go with solid and do targeted resequencing with solid in that area because it is so accurate. However, technologies like solid are not recommended for tasks like de novo sequencing or large genomes, because you need a lot of throughput in order to sequence large genomes and be able to put a genome back together. Like I said, the ion torrent is very low throughput, but it's a good choice for small labs, small projects, It is very fast and does have higher quality reads than Illumina at 100 base pairs in length. And then you have other technologies like Sanger, Roche, and other third-generation technologies. They all have their own application-specific advantages. So just know what your application is and understand that Illumina is not the one answer that can do everything, although it is a great first choice. So these are the learning objectives. Does anybody have any questions, or did I not cover some of these learning objectives? Okay, online question. Would you equate nanostring to the solid sequencing? I might have to do a bit of research on that. I've heard of nanostring, but I'm not super familiar with its actual methodology. So I can look at that and get back to you. Okay, let's see. And we're just about done. So if you only remember one thing, hopefully this presentation got across that knowing how your data was generated can help you choose the right analysis method and also identify errors or problems with the data. I find a lot of people that are just getting into bioinformatics, they get that Excel sheet of numbers and go, great, okay, now let's do stuff with it. Wait. How was it generated? What technology? You need to know those kinds of things so that you don't make improper assumptions about the data that you actually have. So, what time is it? Oh, I got a couple more minutes. I actually wanted to show one example real quick of how the differences between microarray and RNA-seq. So this is actually an experiment, a project that we're doing on the Arabidopsis thaliana plant. And we're looking at ARF19 signaling and how that controls root growth under stimulus through the auxin hormone. And so this has been like a 10-year project. It's still going on. But we originally measured gene expression with microarrays, and the wild type, which has the ARF19 transcription factor active in it, we measured that expression for this ARF19 gene, and we have a fold change of 3.73. And then we went back and did the same experiment, only we did it with RNA-seq, and we did a wild type, which is equivalent to the microarray, so we get about the same expression change for ARF19. But then we did it with RNA-seq when we knocked out ARF19. So this ARF19 gene should have been knocked out. There should have been no expression at all. But the RNA-seq measured an upregulation of 2.82. Now, if this was a microarray experiment, we would not have been able to go down and further investigate why are we getting ARF19 expression. It's knocked out, it shouldn't be there. But because we have RNA-seq data, we can actually go and look at exactly where each read aligns to the genome. And so this is the wild type data, and the height of the histogram is the depth of coverage for that area for this gene. So we can see this is the whole transcript of ARF19. We can see in the wild type, all of it is expressed. However, in the ARF19 knockout, we have expression only at the very beginning. So the stream is actually transcribed in the opposite direction, so it's transcribed this way. So transcription starts here, and so we have expression here, but then none of the rest of the transcript, none of the rest of the gene is expressed. This was actually because the way they knocked this gene out was an insertional knockout. So they inserted a series of bases at this point in the gene. So they knew exactly where they inserted that. That knocks that transcription factor, or knocks the protein off of the DNA or off of the RNA. So you cannot transcribe the rest of it. So being able to par down and dig deeper into RNA-seq data was able to tell us, hey, the knockout actually did work. This is not a functional transcript, so we're good. So we were able to continue with the experiment. If this had been microarray, it would have led to a bunch more investigation. Hey, did the knockout method actually work or not? But because we had RNA-seq data, we could actually look down into more detail. to see that the knockout, yes, it did indeed work. This is just noise. Okay, so if anybody has any questions, I can hang around for a bit. If anyone online hadn't already typed in your name, please do so that I have that record for attendance. And thank you, everyone.